esteemed faculty members, distinguished alumni, valued students from all over the nation. It is with immense joy and pride that I extend to you a heartfelt welcome to the 15th edition of Illuminati, the annual undergraduate medical conference of the Armed Forces Medical College. What began in 2010 as a visionary initiative has over the past decade and a half evolved into one of the most esteemed and impactful platforms for undergraduate scientific discourse and medical innovation amongst medical colleges. We are deeply honored this year to host over 1,300 participants from 58 premier medical institutes across India to embody the ethos of Illuminati, the spirit of curiosity, knowledge sharing, and lifelong learning. Central to this academic fest is the CISO, the Student Scientific Society of AFMC, whose unwavering dedication, meticulous planning, and execution have consistently elevated this conference to newer heights. This year holds special significance as we mark the inauguration of the UG Research Hall of Fame, Anusandhan, and also two pioneering initiatives, Akarjan, the 3D printing lab, and Anveshan, the tinkering lab, both made fully operational within a few weeks. These state-of-the-art facilities are catalysts for innovation, providing our cadets and young researchers with the tools to transform ideas into impactful solutions. In addition, the clinical quizzes of the Student Scientific Society have further elevated our academic profile by organizing 3 national-level quizzes, namely on tuberculosis, multiple myeloma, and diabetes mellitus, engaging participants from across the country. The active participation of our cadets in prestigious forums, such as the International Conference of Military Medical Schools and national platforms like the Indian Myeloma Congress, stand as a testament to their academic excellence at both national and international events. We reaffirm our dedication to collaborate, to uphold the highest standards of scholarly pursuit, and remain unwavering in our service to science and to humanity. May the glow of Illuminati continue to guide and inspire all of us. Thank you.
Thank you so much, sir, for inspiring words which truly illuminated our minds. Moving on to some exciting news, our alumni have once again showed their incredible support.
Firstly, we're beyond thrilled to announce that the L batch has donated 7.24 lakhs to CISO to sponsor the hackathon events in all the subsequent Illuminatis here at AFMC.
We request the L batchers in the audience to kindly hand over the check. Today, we take a moment to remember someone who truly shaped the future of pediatric medicine, late Brigadier Vipin Chandar, who was a mentor to countless doctors, shaping their careers and hearts.
In his honor, his family has generously donated Rs 6 lakhs to SISO to create an award fund for students excelling in pediatrics quiz.
And starting this year, we're also launching the Brigadier Vipin Chandar Memorial Pediatric Undergraduate Quiz, where the winners and runners-up will be recognized in his name.
We request the head of Department of Pediatrics to join us on stage to present the check on his behalf.
But that's not all. The G2 batch has also generously donated 3.84 lakhs towards case presentations, and we request the G2 batchers to come up and present the check.
Lastly, Team Illuminati 2024, moving in the steps of those before them, have also contributed a sum of 6.5 lakhs.
We request the officer in charge and Team Illuminati 2024 to kindly present the check.
We're proud to announce that the Student Scientific Society is the first club to launch a website with its very own e-museum, a digital time machine that walks you through the roots of research here at AFMC.
From grainy pictures of first-generation AFMCites to the humble beginnings of research, it's a tribute to where it all began, when research was more passion than protocol. We now cordially invite our beloved Commandant to unveil the website along with the e-museum.
While we are showcasing our countless achievements, how can we look past the SISO Chronicles?
Introducing SciSo Chronicles, our very own tri-annual time capsule of brilliance. It's not just a newsletter, it's the soul of the club in print.
Packed with articles from cadets and officers alike, it covers everything, from scientific milestones to behind-the-scenes chaos. Whether you're into research highlights or just here for the club gossip disguised as proceedings, Chronicles is your go-to.
We now request the dignitaries on stage to kindly launch this year's edition. This year, we've had the most abstract submissions ever, with a whopping 289 papers submitted for presentations, all of which we've copied and developed into a CD.
This CD will be available soon online on our newly released website for everyone to access. May we now request the dignitaries to kindly launch the CD.
Additionally, Team Illuminati is honored to present an unparalleled celebration of valor, resilience, and innovation, an inspiring call to join the armed forces with our Join the AFMS campaign. Now, we invite Medical Cadet Celine Raphael, Secretary of Student Scientific Society, for the formal vote of thanks.
It is both an honor and a deeply humbling moment for me to stand before you today to deliver the vote of thanks as the Secretary of the Student Scientific Society and the Organizing Secretary of Illuminati 2025, AFMC's annual National Undergraduate Medical Research Conference. Illuminati 2025 was a dream months in the making, and today's inauguration marks a significant milestone in that journey. At the outset, I would like to express our deepest gratitude to our Director and Commandant, Lieutenant General Pankaj P. Rao, whose presence and guidance has propelled our conference to new heights. I would also like to thank our Dean and Deputy Commandant, Major General Kiriraj Singh. Sir, your unwavering belief in the caliber of your cadets has been instrumental in making Illuminati 2025 a reality. A very special vote of thanks goes to Brigadier Suman Kumar and the Officer in Charge, Students Scientific Society, Colonel Vayodhar. Sir, words fall short in capturing the depth of our gratitude. You stood by us through every challenge, offered reassurance every time we faltered, and pushed us to aim higher at every step. I also extend a sincere and heartfelt gratitude to all our HODs and our esteemed faculty members. Encouragement and presence here today have added tremendous value to the conference. Your mentorship forms the foundation upon which we grow as young academicians. I would also like to sincerely thank Brigadier in Charge Adam, Commodore Adam, Colonel Training, the PMC of the Officers' Mess, the Quartermaster, and the Company Commander for facilitating numerous administrative and logistical processes behind the scenes. I would also like to thank the Headquarters Southern Command and all its affiliates for facilitating the Join the AFMS campaign. A special word of thanks to all the tech support staff, maintenance teams, and support personnel who worked tirelessly behind the scenes and ensured that every venue was clean, every setup was in place, and every request was met with a smile. To my incredible H3 batchmates, especially the core committee, thank you for pouring your heart and soul into every detail. Your grit, efficiency, and teamwork have been the heartbeat of this conference, and I am truly honored to have worked with you all. To our junior batches, your enthusiasm and energy and eagerness to help have been heartwarming. You've carried out your responsibilities with such diligence that have made us so proud. I would also like to thank the press and media for taking time out from their busy schedules to cover this confluence of youth and medical research. Finally, to all the 1,342 delegates from various medical colleges across the country who will be attending this conference, thank you for being a part of this moment. With that, I invite you all to immerse yourselves in the scientific spirit of Illuminati 2025, igniting prodigious minds. Let the next 2 days be a celebration of innovation, intellect, and inspiration. Thank you, and Jai Hind.
Thank you, Madam Secretary. As we set the stage for the keynote address of the day, we humbly request Brigadier Suman Kumar to stay on the stage and the rest of the dignitaries to kindly take their seats in the audience.
Today, we're going to have a phenomenal talk on one of the most modern therapies, the gene therapy and the CRISPR-Cas9 technology, which has been brought to developing countries like ours, where they're going to be indigenously patented, thanks to none other than our speaker of the day.
And to introduce the topic, we'd like to invite Major General P.K. Sharma, Commandant AICTS, our co-chair for the day. Sir, an alumnus of the W batch, is an astute physician and gastroenterologist.
We request Sir to join Brigadier Suman Kumar on stage.
Hello. So myself, Brigadier Suman Kumar and Major General P.K. Sharma are deeply honored to be sitting here, chairing this keynote address. And it's my singular honor to introduce Dr. Debojyoti Chakravarti to all of you. Dr. Debojyoti Chakravarti is a leading expert in genome editing and RNA biology, currently serving as a principal scientist at CSIR Institute of Genomics and Integrative Biology. He earned his master's in molecular biology from West Bengal University of Technology, Kolkata, and a PhD from Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany, under Frank Buchholz. After postdoctoral research at TU Dresden, he established his lab at IGIB, focusing on embryonic stem cell biology and CRISPR diagnostic. His work includes developing Feluda assay, a CRISPR-based tool for detecting genetic disorders like hemoglobinopathies, showcasing his clinical potential. Dr. Chakravarti's research also explores therapeutic genome editing for genetic disorders and stem cell-derived organoid models for disease modeling. He has received the CSIR Young Scientist Award 2020 and is an EMBO Young Investigator, reflecting his scientific excellence. Beyond science, Dr. Chakravarti is an accomplished sitarist, performing widely in India and abroad, blending art and research. So his keynote on CRISPR-Cas in medicine will inspire the medical MBBS students who are here, highlighting its transformative potential in healthcare. I request Dr. Chakravarti to please come on the stage and deliver his address.
So good morning, everyone, while this gets loaded. So first of all, it's an absolute pleasure to be here, all the dignitaries who are present here. My only connection with AFMC is a failed medical entrance exam, okay? That's almost 20 years back or so. And at that point of time, I just wanted to visit this place and see how this is, because we used to grow up with knowing about this great medical colleges of India, and AFMC is right there at the top. And of course, I didn't make it, but and that's why I went into the natural sciences, the BSc, MSc, PhD. But I'm just glad that I finally got a chance to come here. Working with a lot of clinicians, so this has been a very special experience actually to come here and see a different setup rooted in so much in service, sacrifice, valor, and of course excellence in medical sciences. So I'm really grateful that I get this opportunity today. So thank you, sir, for the very kind introduction. In case you don't understand the CRISPR talk, there is an easier talk which will be after the break, so you should not go. Okay. So I'll talk a little bit about some of the work in gene editing, which we are trying to do over here. I'm told that most of you are undergrad students. So with a quick show of hands to help me understand, how many of you have heard and know about gene editing and CRISPR? Okay, I only see at the back, so which means I have to explain a little bit what this technology is about before going into details. Okay. So, I come from an institute in New Delhi, which is called Institute of Genomics and Integrative Biology. And this institute was, it was entrusted with sequencing the first Indian human genome, and that they completed in the early 2000s and so on. And then also went on to do the human genome sequencing of other neighboring countries, Bhutan, Malaysia, Sri Lanka, and all. And we got a lot of sequence information. So for the quiz enthusiasts, the number of letters are in high billions, the ATGC letters in the human DNA. So once they knew what are the alphabet in the Indian human DNA, or for that matter, human DNA, they also found out that there are many mistakes in that. So the technology that I'm talking to you about is about correcting these mistakes. And for a long period of time, it was felt that you are born with whatever is there in your DNA. You must have heard that DNA is the blueprint of life and so on and so forth. And there is no way to change this once you are born. How would you change that? But thanks to some of these almost technologies, which has really changed the paradigm of how we look at biology and diseases and therapies, it is now even possible to do these kind of changes. And it's not a holy grail anymore. So much to the extent that we have in the world right now people like you and me who are walking about with their DNA altered. So that's the power of gene editing per se. So what we are trying to do here is to correct errors in the DNA. And I will tell you how this is being done. And I'll also tell you a little bit today about the talk that I'm going to give is not just about the science per se. The science is, of course, great, and you keep on improving things. But sometimes we have to think beyond what we really develop in the lab and think of how we can make it possible for patients to have it. You're all, doctors and, going to become doctors. As long as science is remaining fixed in the lab and there's no way to translate it, sometimes it loses its stigma, sometimes it loses its power kind of. So how to make these things affordable, accessible for people across the country and the world. So that's also something that I'm going to talk about today. So, like I said, that it is no more a holy grail. There are patients who have been treated with, for CRISPR therapies, gene editing therapies, for a disease that you guys have studied. I have seen some sickle cell anemia camps in the first, part of the presentation. So sickle cell anemia is a disease which has actually been cured. When I say cured, it's a very big word in medical, because you technically cannot be cured, your symptoms get away. But here the genetic basis of the disease has been obliterated, which means that the patients are actually fine. They do not have any, let's say, remnants of the disease. So there are patients who have been cured in the US, but then this got approval only in 2023-24. It's not a very early, long period back. But as you see in this article in Nature, I've highlighted here, this one-time treatment is very costly. You have an idea about how costly this treatment? It's a one-time because you're changing your DNA, so essentially you're cured for life. But this treatment costs close to about $3 million per person. So that's about 20 CR, okay, 20 crores to pay to be free of a disease, okay. Now imagine, where do the largest number of sickle cell patients in the world come from? They come from Africa, and followed by India, actually. So if you had to make such a life-saving treatment possible, our patients will never be able to afford that. And you will be very surprised, or maybe you know this already, that most of the sickle cell patients in India, they come from tribal belts. Really the poorest of the poor people. Maharashtra is a huge sickle cell belt also, for example. So therefore, the job that I am trying to do here in IGIB is to see that not only make such therapies possible, but also how to make it accessible. You know, and that's a very complicated puzzle. Really, science is very easy to do in the lab and make therapies, but to make it available to people, that's a completely different domain. It's more complicated than getting the Nobel Prize, probably, because there's a lot of different areas how you have to tackle that. And I will tell you also why this is so costly. So these two individuals, you might know them. One of them definitely is Jennifer Doudna on the left. She won the Nobel Prize, for gene editing in 2020. It's a fantastic, scientist from UC Berkeley. And Fang Zhang, who is from MIT. Now, why I mentioned these, because they were, they were fighting for a long time to have the patent rights over CRISPR. When you have such a path-breaking discovery, you want to own that. And UC Berkeley and MIT had been fighting over this for a long period of time. Who owns the access and who owns the royalties, essentially, to get CRISPR, for CRISPR, which means that anytime anybody in the world uses CRISPR for some application, they will profit from it. And what happened was that through a lot of litigation and legal issues, finally, Broad MIT actually got the patent for CRISPR just a couple of years back. Now that's okay, because as you might know, except for those landmark things in insulin where patent rights were waived off and things like that, most of the medical technologies or developments are actually patented, because it also provides a framework for protecting your intellectual rights and so on. So it would have been nice had this been as simple as that, and one could actually start therapies elsewhere. But if you look at how the Broad Institute MIT tries to, give these rights for making therapies, it's through a very complicated process. So if you go to their website, you will see actually how they make this available for different countries, different patients, and so on. And this is through this very complicated network where they have their own companies. And if you, supposing today you want to make a gene therapy for, let's say, breast cancer, BRCA1, you want to correct the mutation and make a therapy like that, and you have a company you want to set up to make these drugs, you would actually have to go through these companies which has been set up in the West, and you pay them millions of dollars of royalty or millions of dollars of license fees till you get actually the possibility of even making these therapies. Okay. Now that's not possible to do for resource limited settings, or for that matter, even in India, a company which will not have very deep pockets will not invest in such kind of drugs. And that's why these drugs will forever stay away. If even in India, for that matter, anywhere in the world, except where there is really the muscle power and the economic power to do that. And there's no way to get through it, because that's how these patent laws are built. That's how the universities profit abroad. And this means that even though these therapies are available, FDA has approved them, they will never be available for the patients who need them the most. So if I could summarize what we need to make this available, one, of course, is the therapy safety, that how safe you make these technologies. We are correcting an error in the DNA. So what does that entail? The pricing has to be regulated in some way. How you can do that regulation? Of course, a very important part is the IP and licensing. Intellectual property. How do you manage to generate intellectual property yourself? And then, awareness, like what I'm doing over here, and also with patients and doctors, that are they ready for such kind of novel technologies and therapies to actually implement. So I'll come to each of them and how we are trying to mitigate here in India and try to see if these are possible to do. So to cut a long story short, this whole technology works on the principle that there is a bacterial enzyme called Cas protein, or a Cas9 protein in this case. This is shown here. And it takes in an RNA as a guide, okay, which is also called a guide RNA. And it can be directed to go to any 20 to 21 nucleotide sequence in the DNA of a cell by putting in information in that RNA that you have to go and bind to that part of the DNA where that sequence actually matches. Once it goes to that site in the DNA, out of 3.8 billion letters, it goes and finds that site. This Cas9 initiates a double-strand break in the DNA. And then once the DNA is cut, you know what happens when the DNA is broken? It is repaired. Because by the time that I started this talk and till here, I would have had 1 million DNA breaks in my cells, which has gotten repaired. Our cells is very, very efficient in repairing. So it repairs that. Now scientists are smart. What they did is that they saw if you put in a small piece of foreign DNA at that site where the DNA double-strand break has happened, you can actually integrate that. Now imagine if there was a BRCA1 mutation, you cut it at the BRCA1 site using this kind of a technology, and then you put in a foreign DNA, which is the correct nucleotide sequence, you can essentially gene correct what was originally a mutation into a normal cell. Now that's a very simplified way of telling all these things. If you have to take it to therapy, it's a very complicated process. Where would you do it? How would you do it? And so on. But what we saw was that this Cas9 that was being used all over the world, it comes from a bacteria called Streptococcus pyogenes. So it's a bacterial defense system which has been harbored for using in human cells. So the Cas9 originates in the bacteria. It was used as a defense mechanism against invading bacteriophage or virus. And we have managed to take this Cas9 and put it in human cells and do the same activity and essentially change the DNA sequence. So when we were working on this, alternative Cas9, so most of the work that I do here is with this gentleman here, Dr. Shouvik Maiti, who is currently the director of IGIB. He's a friend, mentor, and a colleague. He comes from nucleic acid biophysics background. Okay, so what we found out that if you take in a piece of DNA like this, and then you incubate that with the Cas protein, what happens is that it cuts. It's an endonuclease, so it cuts the DNA. It makes two bands like this because it will have a bigger band and a smaller band. It cuts in the middle. If you do that with Streptococcus pyogenes Cas9, or if you do that with this novel Cas9 called Francisella novicida Cas9, which is from a different bacteria which we are studying, it always does this cut. But interestingly, whenever you take another piece of DNA which has got some mismatches or a mutation in them, let's say 2 mutations, 3 mutations, or whatever, whereas Streptococcus pyogenes Cas9 would cut these as well, we call these as off targets in the DNA because they're really off targets, okay? FN Cas9 would not cut these. Now that's very interesting because that tells us about an important thing called specificity. You know, where your drug molecule should only act on the substrate that it's supposed to act and not on something else which is very similar. That's called a specificity. And why is specificity important? Because it ultimately leads to safety of the drug molecule if you put it inside the human body. You only want to cut the BRCA1. You do not want to cut P53, right? So that would be a problem if that happened. So we thought, okay, that's very interesting because, this Cas9 has actually entered clinical trials, whereas the Cas9 that we are working with seems to be very, very specific in terms of what it is doing. And we realized, you don't have to go into details about these graphs and all, but just to suffice to say that we did a lot of protein DNA binding assays to understand that this Cas is so specific because it can, whenever it sees a mismatch, with the RNA and the DNA, it evicts from its off target. It doesn't bind to the off target anymore. It just comes off. But if it finds a complete match, then it sits there and it cuts the DNA. So that is essentially being told over here using binding affinities and so on. So that was something interesting because we thought that if you give this piece of Cas9 a piece of like a DNA which has got full match with the intended sequence, and you give this another piece of DNA which has got a single mismatch, then this Cas9 should be able to tell what is a full match and what is a mismatch, right? Because it has so much of sensitivity towards a single mutation. So if we could convert all that into a kind of a readout, then we could sequence a mismatched DNA without the need for sequencing per se. You can use Cas9 to do that. So if you remember, when you do sequencing, DNA sequencing like Sanger, NGS, you're actually reading the DNA and you're telling if there is a mutation in that. And doctors often use that to understand if the patient is suffering from a disease, rare disease or genetic disease and so on. So this was possible, and this is actually the origin of this technology which we call as Feluda. Okay. So before I go on, are there any Bengalis in the group here? Yes. Okay, there are 4 or 5. So you might be knowing who Feluda is and what that entails. Just on 2nd of May was the birthday of Satyajit Ray. You know, Satyajit Ray is a big icon in Bengal. And Feluda happens to be a detective character which he built, made in movies and stories and so on. Feluda actually is the world's first CRISPR-based paper strip test for detecting point mutations. Okay. I told you why it was made that way, because the Cas9 can do that detection and so on. And why we kept the name Feluda was because during COVID times, when all research was stopped and the government said you make some kind of a detection for COVID and all, we were kind of competing with 2 other groups, and those 2 groups are the ones that I showed in the beginning, Jennifer Doudna's group and Feng Zhang's group, and they gave names, one based on Cas13 called as Sherlock, and the other one based on Cas12, they called it Detector. So we wanted to give it a Desi detective's name, okay? And since we were both Bengalis working in the team, we gave it Feluda. That was the origin of the Feluda. And we conceptualized this in somewhere in January 2020, and this got approval from the drug controller in September. So it's just about 6 months, 7 months to develop this test. Essentially, I won't go into details, but we married a little bit of CRISPR biology and paper strip chemistry. We made it like a paper strip test completely, like a pregnancy test, essentially, where you can detect through this appearance of these bands whether a sample is positive for COVID or a positive for, or negative for COVID and so on. And these were, things that we could develop to help the clinicians to understand whether this test is positive or not. Topshan, Jatayu, or characters from Feluda stories and so on. But really, we actually made this not for COVID. It was some kind of a thing that came out separately. We actually made it for sickle cell anemia, because that's where a point mutation has to be detected quickly on the field without needing these samples to go to a Sanger sequencing. And sometimes our patients told us that they coming from tribal belts, they would come give their blood like you see over here, and then it goes to, let's say, the nearest Sanger sequencing thing, which is in CCMB Hyderabad, for example, and then it will take almost a month for the results to come in. By that time, the patient would have gone. So we wanted something where these kids could simply, spit into a small tube. The DNA will come out, molecular biology techniques to break the cell. DNA comes out, you do a CRISPR test, and within 2 hours, you will know what a Sanger sequence can do. So that's what we had developed, but then, we had to make it for COVID. And then, if you can play this movie. Yeah, so of course, we are an academic institute, so we had to partner, we have to do the technology transfer. So we transferred this to, I think this has gotten probably once more. Yeah, we transferred it to the Tatas, the house of Tatas. They made a new company called Tata Medical and Diagnostics. They ramped up this whole thing into like a robotic system, put it behind trucks, and then they went to all the places where qPCR machines were not available during the pandemic and so on. And that's what they did over there. That's all fine, but it's more like a distraction because you had nothing else to do during COVID, so we had to put everything in the diagnostics. The ideal place would be to see if we could do gene editing with high specificity. Of course, Feluda is not dependent upon the disease. You can take any other disease. This is, for people in the gastroenterology department. This is work that we did with Dr. Govind Makharia at AIIMS, where he said that in our OPD, people come with, clarithromycin resistance phenotypes, which we cannot do without doing a 5-day culture of these kind of samples. So you could do this using Feluda also, just quickly within an hour or two, to detect if this bacteria is present or not. This came from the northeast, where in Arunachal Pradesh and so on, where rotavirus is very prevalent. And again, there also in the pediatric department, they do not have mechanisms by doing this kind of very quickly without having to culture this bacteria in plates, 3 days, 4 days, and so on. So we made these kind of tools to actually do these tests using CRISPR very, very quickly with a quick turnaround time. But like I said, that the main approach is not to make it for diagnostics. If you have something which is so specifically binding to target DNA, ideally you should be able to cure a disease with that. Make corrections, genetic corrections. So cut a long story short, what we did then is that we had a wild type Francisella novicida Cas9, which was quite efficient, quite specific, but it was not very, very robust in doing the gene editing at the site where it was supposed to do. It's a wild type enzyme, okay? So we collaborated with a Japanese group, and we figured out that if you make different mutations on this protein. So in a protein, there are amino acids, right? If you change amino acids, then you change the properties of how this enzyme can interact with DNA. So if you make very directed changes in these amino acids, you can actually make the wild type enzyme perform many, many times better than actually so we call this as engineered Cas proteins. So that's what we did. So it now became very efficient, and it was even more efficient than the ones that have gone into clinical trials, Streptococcus pyogenes Cas9, without losing this high specificity inside cells, even towards one mismatch of targets. Okay, so one base pair mismatch in the genome out of 3 billion letters, it is still able to distinguish that because of the specificity. And we could show this in different types of locus and so on. Strikingly enough, if we take one of the most common locus here, which is for sickle cell anemia, and you look at all the chromosomes where this can go and bind, if you compare it with the one that went into the FDA approved trial, you would have close to 1,300 off targets across the chromosomes. Now this is very dangerous, because if a Cas goes and binds and cuts to other chromosomes, it means that these might lead to chromosomal breaks. And sooner or later, there could be some chances of having some kind of a chromosomal break related problems, which could end up in having a cancer phenotype. What we saw was that with these engineered Cas proteins, we were having almost undetectable off-targeting, except for that on-target here, which is the HBB gene, which has the sickle cell mutation, for example. And we validated these in patient samples, and we saw that it was very, very specific. So that was something that is very, very unique. And only because of that, we could actually make, second generation, third generation CRISPR-Cas systems. I'll just show you one example of that, where this is work done with the LV Prasad Eye Institute, and Dr. Indu there came and said that, we have a patient here who's completely blind, 29 years old, and that happens because this patient has got a single point mutation here, this TAG, which is the A, which is the mutation, and that caused this disease called Leber's congenital amaurosis, okay? And is it possible to correct that mutation back to normal? So we made what is called as induced pluripotent stem cells from the patient's cells. So we take the fibroblast, skin fibroblast, convert it into stem cells, and then converted the stem cells into the retinal cells, okay, by differentiation. And when you do that and you do a gene correction in there, this is a very beautiful thing to see, that a protein which got lost from childhood is actually restored back to normal. So this means that if you give these cells this retinal patch and put it behind the cornea, which is exactly what these guys are trying to do now through a trial, you could restore vision. Typically, these things are done by finding a matched donor, and make that and putting it behind. But then what happens with matched donor, as most of our clinicians know, is that you need to be very, very careful about immunosuppression and the GVHD and things like that. What we are talking about here is your own cells gene corrected and put back to you. So the GVHD doesn't exist anymore, okay? Or to a very, very minimal extent. So this is something that they're taking ahead for a clinical trial as a possible route to correct blindness, for example. And of course, once we had all this gave us a very unique opportunity. We got an American patent on it because what we realized, and I showed you the first point, was the IP and licensing. If we keep on relying on technologies that we have to import from outside, then we will have to pay all these licensing fees and regulatory fees. Unless we have our own IP from India, the companies that work in this space will not have the flexibility and the freedom to operate, or the FTO as they call it, to start making these drugs. So this was something that we were able to solve using this R&D. And like I said, that my only connection with the armed forces was because when I was young, we used to watch this in Doordarshan during the Republic Day, but through science, I would say, we got an opportunity to be there as a guest at the Kartavya Path last year and see this live, and it's a tremendous experience to see that as a guest from the government. But that's all fine. You can make patents, you can sell technologies. That doesn't solve the problem of what we're trying to do, which is to make gene therapies possible in India. So that's a different science altogether, and I'll come to that now. And like I say, that the path to the clinic is very complicated. You can keep on doing your R&D all your life. You can write papers, publish great papers and all. But the moment you have to take this and put it really in a patient, or to go through the hurdles that are there, regulatory and otherwise, you will realize that what works in the West doesn't necessarily work over here. It's something that is very, very important to understand. You have this path-breaking technologies curing people, but the moment you try to take it and adopt over here, you realize that the Indian system is so diverse, so complex, you simply cannot do that. And sometimes you have to make these kind of adjustments, where the patient is, it's like a traveling paradox, right? You are inside the train and outside the train at the same point of time. Okay, you are traveling regardless. So we have to solve those issues as well. So sickle cell disease is a very important disease from the context of the government. One, because it is, they are rampant across all the countries. Maharashtra is not very, colored over here, but I tell you, it's a huge belt of sickle cell. Most of my calls actually come from doctors. Sorry, maybe I pressed something. Yeah, so it's a problem which is there largely because you're dealing with patients who are coming, like I said, from tribal areas, really very poor people, and the standard line of treatment, as most hematologists know in sickle cell, is to give hydroxyurea. Okay, hydroxyurea is also not available to most of these patients. So imagine telling them about a gene therapy which is going to cause gene correction and cure the disease. So that's almost like a blue sky kind of a solution for this disease. We'll wait for this to come. Maybe. Yeah, sure. Okay, I'll just quickly go to that slide. Okay, thank you very much. Yeah, so to understand more about how the disease context is, I would encourage you to read this very nice article that came in CNN last year, where the reporters actually went with us to the villages to understand how this is being developed. But essentially to tell you that this was a problem which was picked up by the government since 2016, and they said that this will become a mission in the country. And as you might know, that in a couple of years back, our Honorable Prime Minister actually launched this sickle cell mission as with the idea of eliminating the disease by 2047. So we know that it probably won't be eliminated, but we'll make strides towards how to reduce the burden of it. And it is actually a big problem. Doctors often say, hematologists say that the Indian sickle is a mild phenotype, without in many cases realizing that it's mild because the organ damage is not actually monitored. So therefore, when you ask them that what is the the age of the oldest patient, it is always in the 40s, because these people, they just die at some point. So now, and there are also other complicated things like they have high fetal hemoglobin, yet they present with a vaso-occlusive crisis and so on and so forth. Now, what we did is we tried to see if you can use this CRISPR technology that we developed to actually do this gene correction. And through many years, we showed that this is really possible. If you take an apheresis product from a patient, and then you make this CRISPR complexes to correct the mutation, you can almost get from 80 to 90% of the gene correction. So in sickle cell, there's this GTG to GAG mutation. It's a point mutation. So if you revert this mutation back to normal, you can essentially correct the disease. So we could do that without causing any kind of defects in other, in what is called as an indels in the DNA, which means that no other DNA breaks are happening. Quite safely, they can be differentiated into the reticulocytes and so on without off targets in the genome. And then these can also go into preclinical studies where mouse models are engrafted and then they can be cured. But that's quite good. These are very possible to do within the lab. The question is it possible to actually take this to the clinic? And something very strange happened in 2022 when the government actually came and said that, why don't we try a clinical trial for CRISPR gene editing in the country, which will be entirely federally funded. Now, typically such a thing doesn't happen across the world because such clinical trials are extremely expensive. They run in hundreds of crores or something like that as a cost, and they are taken up by companies, by investors, and in the West, that's how it is typically done. But nobody would come and, a company would come and do this for tribal patients because there is no return of investment. So the government said that, okay, we are going to fund you for this, whatever is required, but can you set this up in India? So we did that, and we call it as Birsa 101. Birsa, of course, is in honor of Birsa Munda, who is the tribal, superhero kind of, with the idea that we should be able to gene correct cells from the tribal patients or sickle cell patients and give it back to the patient. These would be hematopoietic stem cells, which would be gene corrected and put back into the body after a round of myeloablation. Of course, when you do such a thing, this is, it's not the, it's way beyond my pay grade when you have so many different, secretaries who have to come together to support that. Because you have a DST which funds, you have a Ministry of Tribal Affairs which looks after the people in the tribal community. You have the ICMR which makes the ethics committees and the national ethics committees for trials. You have the DBT which gives the RCGM approvals. You have the CDSCO DCGI, which gives you the approval to test a new drug and a clinical trial. And CSIR, of course, which is my parent thing. So it was only possible because at some point, Niti Aayog said and came and said that, okay, we are going to do all this for you. So now Niti Aayog monitors it and they bring all the secretaries to the same table. And then we can figure out how to go for the next step. So this has been a truly kind of a collaborative effort, I would say, across all the possible departments that could be involved in this. And to do that, we had to actually set up a completely new GMP facility. Because imagine you have a patient's blood coming in, and you have to take CD34 hematopoietic stem cells out of that, do a gene correction, and do this all aseptically without having any kind of contamination, so that the patient who gets enrolled in AIMS, and AIMS had to build a ward for these patients there, and they have to get this with that high safety that there is no contamination during this whole process, and then the patient has to be followed up for any kind of crisis and engraftment. So all this had to be set up. We are currently at the preclinical phase, finishing across the regulatory, and if everything goes fine, perhaps in the next year we might have the first patient dosed. So the last part of the talk, I can talk about details more later on because the time is running out. The last part of this is to tell you that CRISPR or gene editing technologies is disease agnostic, because it's essentially correcting a genetic defect. So a large number of genetic diseases fall under the rare disease category, where you have no possible cure because they are point mutations or mistakes in the DNA, which there are no drugs for. So, and several of them come in the rare category, and in a country like India, a rare is not a rare because of the population size. There are thousands and thousands of patients who suffer from them. So I'm going to tell you a story about such a rare disease patient. So there was this couple who came to us in just after COVID in 22. They said that their daughter was suffering from this ultra-rare disease. So within rare, there is a category called ultra-rare, which has extremely few individuals across the world. And the mutation that was presented here is this G here is getting converted to A in the SERPINE protein. And what happens when that mutation happens is that the protein tends to aggregate in the neurons. And because of this aggregation, there are seizures, there are epileptic seizures, slowly leading to loss of all sensory perceptions, whether it is vision or smell or anything. And then the patient dies in their teens. So this was a 17, 18-year-old girl who presented with this kind of disorder. And there's no cure, absolutely no cure for this at this point of time. So this parents, they were quite well-to-do. They were funding New York University Langone Center, which is a very top center for schizophrenia and other diseases, and to develop, because they know that there is a gene therapy in the horizon. You could correct this kind of genetic mutation, and they were funding this. But they said that if you try to do this in the US, the regulatory process is so complicated that you're not going to get a drug before 6 to 7 years. And neither is the cost going to be possible to be supported by just a family's, savings, for example. So then they came and met us and said that, would you be able to take this up? So quite naive as we were, we took this up. I won't do that again. But essentially, we had to develop a CRISPR therapy from scratch to correct this mutation, okay? And we did that. So we made a CRISPR, proteins which would actually correct this mutation back to the normal in 80% or so of the cells, which was clinically that's enough. So you don't need to correct more than that. But more importantly, we took this patient's cells, the saliva, because she was already drooling when she came, so we could not take blood from her. So we took that saliva and we converted it into stem cells. There is something called reprogramming, as you might know, also won a Nobel Prize. Through that, you can take any body cell and convert it to a stem cell, like the embryonic stem cells, without having to go to the embryo. So we reprogrammed that and we differentiated that into neurons. So we made neurons on the dish with that. And we saw that upon gene correction, you could actually reduce this, aggregation back to almost like normal levels. So pathophysiologically also, there was a benefit you could associate. We did the whole genome sequencing of this patient to see if there are any off targets that this CRISPR therapy might have. We found there was only one off target here, but it was lying in a gene desert, meaning there's no gene over there. So you would not hit something by mistake and cause some problem. So all that is good because you have a CRISPR therapy which can correct it. But then you have to put this in the neurons, right? So you cannot simply take a Cas9 protein and put it in the neuron. So there are technologies which are based on viruses. And some of these viruses are called adeno-associated viruses, which are quite, let's say, partially safe for gene therapy because they have been used for gene therapy. These viruses can package this kind of a CRISPR material and can be injected into the body of an individual. And these viruses can be pseudotyped, meaning that their capsid proteins can be changed so that they only go to neurons and not to the liver, to the heart, or so on and so forth. So we had to package all this in a virus, and I'm not a virologist, so I collaborated with one of the top virologists in the country who's making these adeno-associated viral vectors in Bangalore at the Narayana Nethralaya Center. And we put all these CRISPR proteins, and we tried out different ways of injecting into the brain in mice. So close to 200, 300 mice in different routes, ventricular, intrathecal, intravenous, found out what would be the immune responses to each of these processes, how much of gene correction we would get, and so on. So all this has to be done if you have to go for a therapy, okay, for a, to see if this is kind of safe and so on. So once all this was done, I wanted to give you a little bit of an idea about the timeline, because I told you that when the patient presented, she was already 17 years old, and typically these patients die in their teens and so on. So it really needed efforts where all possible stakeholders from across the world actually came to support this. So we had Oren Devins's group. He's really a big shot in the field of neuropathologies, who gave us his reagents. We had only one single scientist in the world who studies this disease, because it's an ultra-rare disease, and she generated antibodies for it. She shipped them all within 72 hours to Delhi, and we started doing all these stem cell experiments, mouse experiments. Within 1.5 years, we finished all this, and then we approached our regulators in India, which is the DCGI, CDSCO. And we told them that, look, there is a patient who has to get this therapy. Can you help us? And they were extremely forthcoming. We had no idea that, that so much of support is there for such kind of rare disease. And they said, look, this is a rarest of rare neuropathy. So there is something called a sunshine trial, or there is an N of 1 trial, where if a person needs a therapy to survive, then we would not ask you to go and do this in 10 monkeys or 20 apes or something like that. So you can go ahead. They gave the approvals, they audited the facility, and we started with the production of AAVs. The idea was that somewhere in 2024, also we also gathered a group of very heroic neurosurgeons who said that what would be the mechanism of doing this injection in the brain at Narayana Nethralaya, and figured all that out. But unfortunately, whatever we tried, in October 2024, this patient passed away. And in August, we had actually met this patient. And when we met this girl, she was already carried to this place with 2 helps. She could not see, she could not eat, she could not drink, she could not hear. She was in a very bad condition. Yet her parents were telling her that there are these doctors here who are trying to develop a new therapy for you, with that kind of a hope. So this talk today is actually in remembrance of Uditi. So Uditi is this girl whose parents actually put all the hope in the basket of science. They wanted to fund and develop a new therapy all over by itself in a country like India, where there is no therapy currently existing in the world actually. And it required, almost, as I say, that it requires a village to do that. We also tried the same way, although unsuccessfully. But you can go and see this article that Nature has made. They had, they wrote about this whole process, which we tried to do in India last year, to understand the depth of this. But it told us something very interesting, is that, Uditi is not isolated. There are many such patients from all over the country who have these kind of ultra-rare pathologies or rare diseases, for whom there is absolutely no hope. And that's the necessity to understand that there is a very long queue of patients. Okay? And time is a commodity for many of them. Like in our field, in the clinical trials field, people continuously talk about safety, efficacy. And one of the prevalent thoughts is that you can make things safer and safer, but at one point you have to give it to the patient to understand really whether it works or not. So you can do hundreds of mouse experiments, thousands of animal experiments, but actually the experiment that you need to do is to have possibility of giving these drugs to patients to understand, especially in the rare disease scenario, whether they're acting or not. And this is something that the FDA is also now slowly recognizing. So you can make these editors, and that's what we're trying to do through science. The pricing, I have told you, the moment we started developing these technologies completely, from their consumables to all the components in-house in India, the price dropped. So right now, we have, the cost, like I said, was $3 million US, which is around 20 CR. the cost that we could make right now is around 70 lakhs, close to what that is that of the CAR T therapy. But the government is not happy. They want us to make it much, much lower than that, because it has to be given almost free of cost to tribal patients, right? So the R&D keeps on moving towards those directions, more indigenization, more reduction of the costs associated with developing the pipeline, and developing or more technologies where we can scale it up instead of having to import from outside. IP and licensing, I cannot tell you how important that is. The moment we got an American patent, we had close to 10 to 15 companies which would have never come up to come and say that, okay, now we have the confidence to start building this from scratch. One of them is also in Pune, to make sure that these can be scaled up to really, make these kind of therapies available in India, both not only for Indians, but also possibly to make it available for other countries as well. Awareness, so I used to think that awareness would be a huge issue because I keep on going to the villages, talking to the people. But they seem to be more aware sometimes than even our doctors, actually. So I had a farmer who met me last time in a village in Bilaspur. He came and said, he was 37 years, he had ulcers on his feet, sickle cell anemia patient. He, Doctor Sahab, मुझे CRISPR के बारे में कहीं पे जानकारी हुई. I want to be a part of that trial. Although we cannot take him because he doesn't have enough HSCs, we need 20 million CD34, something like that. But that just tells you that the patients are well aware of the technologies that are there. And it's a kind of, we have to work together along this, the doctors. So we have a very good team in hematology AIMS who is actually leading the sickle cell trial, and then doctors elsewhere as well. So yes, awareness is as important as well. So this is the most important slide. When I talk here, I'm just, selling the science, nothing more than that. There's a very big team which works with me. There's very dedicated students. I found excellent students here also. If you have a calling to do gene therapy, gene editing, something that you would like to also take up, please contact me as well, because I think the combination of rigor, service, and medical brilliance that you guys have here is something that is needed for almost like, the DGICMR calls it like a Chandrayaan project. In a way, it's a Chandrayaan project because, we are dealing with a lot of different complexities to take this for such patients. But please, you can reach out to me and we can see how we can work together. But there is a lot of people from all around the world. We are very generously funded by different sources, particularly Niti Aayog's help has to be, and DGICMR's help also has to be mentioned over here. Otherwise, such kind of efforts will not really bear fruit. So we are not there yet. We are trying to be there, and hopefully with the support of everybody, we can cross over the line. So I thank you once again for this invitation to come and speak to you about this topic. And I don't think so there is a question answer now because it's an oration probably, but in case you have, I am available throughout the day, so you can reach out to me. Thank you very much.
Thank you, Dr. Debjyoti. That was a masterly oration on a complex subject, and right from the mouth of the expert gene editor or gene character, I'll say gene character,
Gene
or
therapy.
gene editor of the nation. And I must also admit that FMC laws has been a CSIR gain for the larger purpose of science in the country. And any questions from undergraduate? You made it very simple, actually. Such a complex subject, made it very simple. And any thoughts from undergraduates, because it's your conference?
Yes.
Please.
Yeah.
Good morning.
Good morning.
So I have a question. So with increasing focus on biotechnology and genetics, as you said, so how exactly will the regulatory framework evolve in the next few days with regards to this treatment?
It's a brilliant question, actually. So, as we are developing these new technologies, we realize that there are actually no regulations for, even things like advanced stem cell therapies, okay? And it's important to have that because you might have heard about the Chinese scientist who edited embryos and then was, had to go to jail and things like that for HIV thing. So new laws have to come in. And I'm very happy to tell you that these are coming in. I'm also in the DCGI CIDISCO board for framing these new laws. And we are coming up with a set of guidelines, which should be, these are guidelines, which is essentially a dynamic thing where we take in inputs from stakeholders to make this in accordance with what is happening elsewhere. Take inputs from all the people, FDA, EMA, and everything. But at the same time, keeping the general infrastructure in India in consideration as well. So these are getting framed, and I think in a month or so, this will come out. These will be guidelines for cell and gene therapy, particularly with emphasis to gene editing and so on. Yeah.
So if there's a question, we thank you, sir, for such a masterly oration. And we applaud you for this in our usual manner. And
And with that, we wrap up the opening ceremony of Illuminati 2025.
We thank everyone from the bottom of our hearts for being a part of Illuminati 2025. Let's continue to ignite, illuminate, and innovate.
Now we request the faculty to kindly join the keynote speaker on the bridge. As the faculty proceeds, let me remind all the delegates and the cadets to kindly remain seated while we set the stage for the next event. We're now going to continue our session with our upcoming exciting event, the Symposia. where minds meet, ideas ignite, and that is the true spirit of a symposium.
With this, we welcome you to Conflare, the medical symposium.
I would now like to welcome the chairs for the event. First up, we're delighted to welcome Colonel Kanchan Kulhari as the chair for today's symposium at Illuminati 2025.
A dedicated academician and clinician, she brings wealth of experience in clinical biochemistry, quality control, and medical education, and has 18 research publications and 5 AFMRC projects under her wing.
Ma'am's work has been recognized with multiple accolades, including 2 Best Completed AFMRC projects and Best Oral Paper Honours at Ambicon 2022 and 2024.
A recipient of the prestigious GOC Western Command Commendation, she embodies excellence in both research and service. We're truly honored to have her chair this session.
Co-chairing with Ma'am is Colonel Deepthi Saharan, an alumnus of AFMC. Ma'am holds an MHA and from AIMS, along with multiple certifications in hospital management and quality assurance.
With 20 years of stellar service in the armed forces, she brings deep expertise in quality assessment, leadership, and policy development. A decorated officer, she's a 3-time GOC Commendation Card awardee and a certified assessor for both NQAS and NABH.
Her passion for teaching, sharp administrative insight, and commitment to excellence makes her presence today truly inspiring.
With this, we hand over the stage to our chairs and humbly request them to introduce our judges for the day.
I thank you, the MCs, on our behalf for the kind introduction. Good morning, everyone. We begin the afternoon with a symposium. All of us know the topic of this year's Illuminati, where minds meet, ideas ignite. So we start off the spirit of this conference, student conference, with the symposium.
Good morning. We first welcome Dr. Dipali Rao. Her previous appointments include Senior Consultant at KPMG and Deputy Medical Admin at KEM Hospital, Pune for a decade. Ma'am is presently working in Bharatiya Vidyapeeth Medical College and Hospital as Head, Health Economics and Strategy. Her experience is invaluable to us and we are privileged to have her as a judge. We welcome you, ma'am.
We also welcome Brigadier Mandeep Saini, Brig IC and Commander Troops, AFMC Pune. Sir is an alumnus of AFMC from the A2 batch, holds an MD and DNB in radio diagnosis, and a postdoctoral fellowship in healthcare administration from TIS, Mumbai. Sir has received COSC commendation. He is an NABH assessor. Apart from being a professor in radiology, his passion for healthcare quality and leadership shines through every role he takes. We welcome you, sir.
We also take pride in welcoming Brigadier Anuj Chawla, Brig Training at AFMC. Sir is an alumnus of A2 batch and he is professor at the Department of Physiology. Sir has done DNB Physiology and MPhil from University of Madras, fellowship in medical education from Philadelphia, USA. Sir has got various awards and sir has also been a fellow at the Royal College of Physicians, Edinburgh. He's also taught at various prestige institutions like University of Madras. Sir has done NDC. Sir, thank you for gracing the event for your presence today.
Moving ahead, it's our privilege to welcome Air Commodore Aditya Sapra, Head of the Department of Anesthesiology and Critical Care at AFMC, a decorated officer and an academician par excellence. Sir holds the Chief of Army Staff and Chief of Air Staff commendations, a testament to his excellence in service. He was also awarded the gold medal for topping MD anesthesia at RGUHS in 2003, and his contributions to the field through numerous publications continue to inspire young minds. We are truly honored to have you, sir.
We also welcome Air Commodore Anurakshit Gupta, Professor and Head of Department of Surgery, AFMC. Sir is a distinguished surgeon who has served across every corner of India and even in Afghanistan and brings decade of surgical excellence to the forefront of medical education. Beyond the scalpel, he is a celebrated quizzer, being the director at the Asian International Quizzing Association, London, BBC Mastermind finalist in 2002, and part of the Trivia Hall of Fame in Los Angeles. You might have seen him in KBC as an expert. He's authored 4 quiz books. A true blend of clinical mastery and intellectual curiosity. We are privileged to have him as a judge for today's session. We are honored to have you amongst us, ma'ams and sirs.
Before we begin, we will just spell the rules for today's event. The rules are as follows. Teams coming on stage will be presenting a topic of medical significance of their choice. Each team can have a maximum of 10 members. The time limit is 15 minutes for presentation and 2 minutes for the questions and answers. And they would be provided with extra 2 minutes for the setup and tech check.
The teams will be judged on novelty, content, original research, audio visuals, poise and diction of the speaker, time management, and the question and answer round. We now begin with the first team for the evening, Team A, with the topic, Bariatric Surgery as a Metabolic Game Changer, Current Concepts and Future Directions. Bariatric surgery, once viewed primarily as a weight loss procedure, is now recognized as a powerful metabolic intervention. It has demonstrated significant effectiveness in the improving or even resolving conditions like type 2, diabetes mellitus, hypertension, and non-alcoholic fatty liver disease. As obesity and metabolic disorders rise rapidly in India, the future of bariatric surgery lies in redefining techniques, expanding access, and integrating it into a broader metabolic care frameworks. We invite the Team A now.
Team A, your time for tech check starts now.
Hello. Hello, hello. Check, check. Hello. Good morning, everyone. We are from Stanley Medical College, and we are here to present on the current concepts and future directions of bariatric surgery, the metabolic game changer. So, increased number of bariatric surgeries are being performed today. And do you know when the first bariatric surgery was performed in India? It was in 1999, and it was in Pune by Dr. Sri Hari Durai Patil. So, these are the topics under which we are going to present. So, first we'll see and understand about obesity before we go deeper into metabolic surgery. So WHO defines obesity as excessive abnormal fat accumulation with a BMI more than or equal to 30 kg per meter square. And instead of just grading with the appearance and the BMI, visceral fat accumulation should also be considered. Visceral fat is the abdominal fat and it surrounds the vital organs such as liver. Its function is to cushion, but it's metabolically active, and excessive accumulation of this visceral fat leads to hazardous health. This causes most of the pathological changes, the complications, and etc. of obesity. So this is measured normally using the waist circumference, the cutoff for which is 1 or 2 centimeter in males and 88 in females. So obesity. The body's energy mechanism runs in a delicate circuit controlled by the arcuate nucleus of the hypothalamus and aided by other hormones such as the gut hormones ghrelin, which increases appetite, and leptin, PPY, which decrease appetite. When there is derangement in this circuit due to environment factors, lifestyle changes, gene-environment interactions, etc., there is obesity. So the complications of obesity are these. First, there is dyslipidemia, type 2 diabetes, and hypertension. All these lead to syndrome X or metabolic syndrome. Next is fatty liver disease, CAD, etc. The other complications include obstructive sleep apnea, osteoarthritis, and all. Next, coming to the prevalence, we can see that the United States has the highest prevalence of obesity. While coming to India, we can see that there is lesser obesity prevalence in India because it's primarily a labor-intensive country. But due to changing lifestyle, India is soon becoming a hub for obesity. So now we'll see into the treatment modalities and how bariatric surgery is the metabolic game changer. So coming to treatment modalities, first there is lifestyle and behavioral changes. This deals with reduction in food intake, increase in physical activity. Then coming to pharmacotherapy, GLP-1 receptor agonists are the latest FDA-approved drugs, semaglutide and tirzepatide. And the older drugs are fixed dose combination of phentermine and topiramate, bupropion and naltrexone. The other drugs include orlistat and metformin. Now coming to bariatric surgery. So baros means weight and iatros means treatment. So this is surgery performed mainly for the treatment of morbid obesity. And this has become the clinical hot topic now for the treatment of morbid obesity because of the studies that show an increased success rate and increased efficiency of bariatric surgery compared to other modalities. And it is called metabolic surgery because it not only causes remission of obesity, but also improves and causes remission of the other comorbidities in obesity, such as diabetes mellitus and metabolic syndrome. So what is the mechanism of metabolic surgery? First, there is restriction and malabsorption due to the surgery itself. And there is hormonal alterations such as decreased ghrelin levels, increased GLP levels, increased leptin levels. And according to research studies, there is gut microbiome changes, which also causes remission of obesity. All these act in the gut-brain axis and change that, leading to remission of obesity. And why did I call it as metabolic game changer? This is because it is not only a surgery which causes an immediate weight loss, and it is not a surgery that is used only for obesity. It causes rapid and sustained weight loss, it causes long-term remission of obesity and its comorbidities, and it causes increased insulin sensitivity, increased and improved lipid profile, and it enhances the energy expenditure. It also alters the gut microbiome and the gut hormones. So, who are all eligible for surgery? Any patient with BMI more than or equal to 40 and anyone with BMI more than or equal to 35 with high risk comorbidities and patients with class 1 obesity with poor glycemic control are all potential candidates for bariatric surgery provided their non-surgical method of weight loss have failed. The absolute contraindications include severe anesthetic risk and pregnancy. Organ dysfunctions and psychosocial instability are the relative contraindications. Other special conditions which are not contraindications but include special attention are HIV/AIDS, extremes of AIDS, and cirrhosis without portal hypertension.
We have to keep in mind that every patient is a unique individual and the surgical care should be tailored according to their special needs. The preoperative care involves a multidisciplinary approach, and there are conditions like obstructive sleep apnea and hyperventilation syndrome of obesity which have to be managed with continuous positive airway pressure and screened with polysomnography. The upper GI endoscopy should be taken in order to rule out Barrett's esophagus, and a routine DVT prophylaxis should be done. Preoperative weight loss is essential before surgery because it reduces the size of the liver and improves surgical outcomes. Anesthetic care, once again, should be tailored according to the patient's needs. Increased BMI decreases the margin of safety of anesthetic drugs and increases the risk of respiratory failure. Poor right ventricular function and increased pulmonary arterial pressure provides a risk for pulmonary hypertension. Airway management is a nightmare for anesthesiologists. in bariatric surgery because of the following reasons. Altered pharyngeal anatomy causes difficult intubation, and a high Mallampati grading like grade 3 or grade 4 causes specific considerations in difficulty in intubation, and a reverse Trendelenburg position is essential during the surgery, and a ramped up position is essential for tracheal intubation. Laparoscopy is the current trend in surgery. That holds true for bariatric surgery as well, because of the following reasons, like decreased postoperative pain, postoperative infections, wound adhesions, and blood loss, and as a faster recovery. The scar pain that is produced in open surgery causes shallow breathing for the patients after the surgery, which might lead to complications like lower lobe atelectasis. Hence, laparoscopy is the choice of surgery. Laparoscopic surgery for bariatric surgery include a large-sized gown, wide examining tables, extra long trocars, extra long staplers are required, and a liver retractor is essential to have complete view of the operating table. We will be discussing about 3 surgeries in detail, starting off with laparoscopic sleeve gastrectomy, which is a purely restrictive procedure, and there is a reduction of ghrelin levels, which reduces the appetite and improves weight loss. There is less complications like internal hernias and malabsorption. The Roux-en-Y gastric bypass is a combined surgery of restrictive and malabsorptive component, and resolution of comorbid conditions. The biliopancreatic diversion with the duodenal switch is a combined surgery also, but it is primarily a malabsorptive one with increased weight loss, and is technically challenging for the surgeons to perform and has significant nutritional deficiency. Now let's look into an animation video of sleeve gastrectomy. The liver is retracted at first, and the greater curvature of stomach is devascularized, 3 centimeter proximal to the pylorus, and a 34 to 44 inch bougie tube is inserted to keep it as a guide, and keeping it as a guide, we will be resecting the stomach from the antrum up to the angle of this by sequential linear firings of stapler, and the greater curvature of stomach is transected and removed. The Roux-en-Y gastric bypass involves in creating a 10 to 15 ml gastric pouch, which contributes to the restrictive component of the surgery. And then the jejunum is divided 40 centimeter distal to the ligament of Treitz, and the distal end of this rulum is anastomosed to the gastric pouch, performing a gastrojejunostomy. And the biliopancreatic limb is anastomosed with the jejunum, performing an end-to-end jejunojejunostomy. Here, the duodenum and the proximal ileum is bypassed, creating a malabsorptive component. The laparoscopic biliopancreatic diversion with the duodenal switch involves in a sleeve gastrectomy as discussed earlier. The greater curvature of stomach is resected and taken out. Now 2 centimeter distal to the pylorus, the duodenum is divided, and 250 centimeter proximal to the ileocecal valve, the ileum is divided. This distal end of the ileum is anastomosed to the duodenum that has been divided by performing a duodeno-ileostomy, through which the foot passes through, and the remaining biliopancreatic limb is anastomosed to the terminal ileum, performing an ileo-ileostomy, through which the biliopancreatic fluids come through. So the fluids are mixed only in the terminal portion of the ileum, creating a heavy malabsorptive component of the surgery.
Okay, we know that bariatric surgery offers several benefits, but it comes with one set of disadvantages. Now let's look at some of the complications of bariatric surgery. Some of the complications which are common to almost all the bariatric surgeries are bleeding, surgical site infection, pulmonary embolism, reoperations, stenosis and strictures. Of these, the pulmonary embolism has the highest mortality rate. Reoperations also common following the bariatric surgery. Some of the long-term nutritional and psychosocial concerns are, there are iron deficiency anemia, vitamin D, calcium deficiency, magnesium and zinc deficiencies are seen. And also, there is an increase in the prevalence of regular alcohol consumption and suicidal tendencies in the long-term follow-up of post-bariatric surgery patients. Some of the complications peculiar to each of the bariatric surgery procedures are, for sleeve gastrectomy, there is a staple line leak, which is most commonly seen along the angle of S, and stricture or stenosis seen along the incisura angularis, and there is an increased symptoms of GERD because of the high pressure tube which is created. For BPDS, the most common complication is protein malnutrition, and the incidence of marginal ulcer has been drastically decreased with the introduction of duodenal switch, which preserves the pylorus. And coming to RYGB, anastomotic leak, small bowel obstruction are very common. And the small bowel obstruction can be detected by contrast central CT, where there will be the contrast cutoff point will be seen, which denotes the point of obstruction. Now let's move on to what are the various benefits offered by the surgery in the comorbid conditions. The first point is that we all know that bariatric surgery is being primarily performed to decrease the excessive weight loss. And we know that bariatric surgery offers sustained a long-term reduction in the weight loss compared to other medicine processes. And when it comes to diabetes mellitus, it causes a decrease in the incidence of type 2 diabetes mellitus in the post-bariatric patients and also remission of diabetes mellitus in existing patients with diabetes mellitus. And there is a 50% reduction in the both microvascular and macrovascular complications. For cardiovascular system, there is reduction in the hypertension and dyslipidemia, where there is a decrease in the LDL and cholesterol level, and also causes increase in the cardiovascular strength. And there is increased symptomatic relief for OSA and musculoskeletal disorder because of the reduction in the weight. And when it comes to OSA, we see that there is reduction in the fat around the neck region, which offers symptomatic relief to the OSA patients. And the additional benefits which are offered in RYGBR, there is improvement in the healing of venous cyst ulcer. We know that reduction in the weight gain, reduction in the very weight leads to increase in the venous outflow, which causes a healing of the venous cyst ulcer.
Now, the surgeries don't always go how we plan to. So the surgeon needs to be prepared to deal with any complications. So in the immediate aftermath, the patient needs to be advised about diet, exercise, and other such lifestyle factors. And the surgeon, again, needs to be on his toes to spot any complications immediately and treat them promptly. And in the long term, we need to monitor the patient's weight, the progression of their comorbidities, assess their psychological profile, and also be on the lookout for any nutritional complications. Now, the very mechanism of the surgery itself predisposes the patient to deficiencies of essential nutrients, right? So surgeons need to routinely screen these patients for any deficiencies, also offer prophylactic therapy for these nutrients, and also be on the lookout for any manifestations. Now, the treatment part is really important here because it can even lead to very serious than weight loss. Now, as we can see, the older band-related procedures, the more